Good morning. I think we can start now. Welcome everybody to today's Connect Science Talk. My name is Ernesto from Project Lab, and today I'm very happy to introduce today's speaker. I was just telling Dr. Racefield that when I start my PhD, I get the opportunity to read his papers about triple-negative breast cancer, and they were so clear and so well explained that I really enjoyed reading them. Dr. Racefield is Chief Experimental Pathologist in the service and Director of the Experimental Pathology Fellowship in the Memorial Sloan Kettering Cancer Center. Dr. Racefield graduated as a medical doctor from Federal University of Paraná in Curitiba, Brazil. Later, he did a training as pathology in the Federal University of Paraná, Brazil, and also in the Institute of Molecular Pathology and Immunology in the University of Porto, Portugal. He received a PhD in molecular pathology in the University of Minho, Braga, Portugal, and the Institute of Cancer Research at the Royal Marsden Hospital in London. Dr. Racefield has received multiple awards. I'm not going to name all of them. Just this year, he has the award with the Goody Medal, from the Pathology Service Society of Great Britain and Ireland, and he also received the BCRF Larry Norton Award for Excellence in Breast Pathology in the International Society of Breast Pathology. His research is focused in molecular pathology of molecular and histological special types of breast cancer and genotypic phenotypic correlations in rare cancer types, which is what he will talk about today. Before we can start with the talk, I just want to remind you that this talk will be recorded, and please type your questions not into the chat option, but into the Q&A option so we can have Dr. Racefield answer your questions. We really appreciate your time, and please take the lead, Doctor.
Thank you ever so much. Thank you, Ernesto, and thank you, Nelly, for the kind invitation of being here with you today. It's virtual, but still we can connect and we can all discuss one of my passions in research, that is the exercise of understanding the genetic underpinnings of these rare cancer types. And what I'd like to do is to persuade you in the next 35 to 40 minutes that studying them is much more than a stamp collecting exercise. So I'm a pathologist by training. Just let me provide some qualifiers before I start. I'm a pathologist. I am a card-carrying pathologist. Then I did my PhD with Alan Ashworth in London. Then, more out of necessity rather than by design, I had to retrain as a computational biologist struck bioinformatician. And the work that I've been doing follows three different areas. One is the one that I'm presenting to you today. The second one is focused on tech dev and development of new cfDNA assays. And the final one is on artificial intelligence applied to pathology, which I will touch upon today as well. So these are my conflicts of interest. So here they are. And in particular for this talk, I need to emphasize that I work as a consultant, and I'm a scientific advisory board member of Page.ai, which is a spin-off from Memorial Sloan Kettering, where we seek to develop AI solutions for pathology. But everything will be disclosed during the talk. So in the next 40 minutes or so, what I'd like to do is first to just recap with you where we stand in terms of the genomics and precision medicine, in particular for breast cancer, and the fact that actually a lot of the diversity from a phenotypic perspective in breast cancer has actually not been taken into account in large-scale studies, and how by bringing pathology into genomics, we have been able to identify novel genotypic phenotypic correlations and even new cancer genes. And then finish this the other way around, how genomics is now informing pathology and helping us develop AI-based algorithms to diagnose special histologic types of breast cancer much more accurately. So I think now in 2023, after the completion of TCGA, PCOG, ICGC, we have come to a point where we have understood what is the repertoire of somatic genetic alterations in common cancer types. We have come to understand even the interplays between germline and somatic genetic alterations in these cancers, and have come to understand that there's a finite repertoire of somatic driver genes, some of which actually are pan-cancer or can be found across many different cancer types. And examples of that's like PIK3CA, KRAS, PTEN, RB1. And there are some genes that are much more cancer specific, even within the common cancer types like VHL. And using all of this type of knowledge, aggregated with the incredible development of small molecule inhibitors, immunotherapy approaches, and also antibody drug conjugates, we have seen the realization of the potentials of precision medicine, in that we have seen the development of basket trials, umbrella trials, and specific trials with biomarkers, basically linking specific genetic alterations with specific therapeutic approaches. It was not the panacea that all of us hoped for back in the late 2000s, early 2010s, but it does benefit a large proportion of cancer patients. Importantly, however, in the context of these precision medicine studies, large-scale initiatives that have been carried out, one point that seems that we had forgotten before we learned it was the importance of the heterogeneity of breast cancers. We pathologists have been aware of this incredible diversity and heterogeneity of breast cancers for many, many decades. And we have actually sought to devise methods to convey this information back to the clinicians. One of the easiest ways that we pathologists use to convey this diversity across breast cancers was in the form of histologic grade. In histologic grade, it's a measure of differentiation, but most importantly, it's also a very strong prognostic marker, and even in some contexts, a predictor of a neoadjuvant therapy response. And grade has now been incorporated into pathology reports around the world, and we use this information actually actively to manage patients with early breast cancer. Grading is applied to the garden variety of breast cancers, the so-called ductal carcinomas, and also to lobular carcinomas. But one point that actually has never been incorporated into clinical practice is the fact that there are many histologic special types of breast cancer. And these histologic special types of breast cancer have different histologic or morphologic appearances. And what I would like to discuss with you in this talk is that they have important biological differences and natural histories as well. Unfortunately, histologic types of breast cancer have never been incorporated into, let's say, the management guidelines, with some, apart from some small aspects incorporated in the NCCN in the last 2 years. And one of the major reasons for that is that, we pathologists are not very objective in the way we diagnose these rare special types. And this is a bit of a catch-22 because there are not many clinical implications for identifying these rare subtypes that are highlighted here. Pathologists do not strive for providing this information in a sufficiently accurate manner. And this has also impacted the way the large-scale studies were actually carried out, to be honest. Let me just organize my screen. Actually carried out. In the large-scale studies carried out by the TCJ, PCOG, and ICGC, actually, these rare cancer types were not included, not in a diligent manner. And this slide here actually was published 10 years ago, or 10 and a half years ago, and still remains current. And it basically shows us the repertoire of somatic genetic alterations that we see in the garden variety of breast cancers. And there are several conclusions that we can draw just by eyeballing this slide here. What you're seeing here is in each column is a gene, each row is a tumor, each tick here is a mutation coded according to the legend here. On the left, we have the molecular subtypes of breast cancer, or the intrinsic subtypes, luminal A, luminal B, HER2 enriched, and basal-like. And on the right, we have the clinical markers like ER, PR, HER2, tumor size, and nodal status. And just by looking at this, we can conclude first that the repertoire of somatic genetic alterations in ER-positive or luminal tumors is vastly different from that of triple-negative or basal-like cancers, in that luminal tumors have mutations affecting PIK3CA, MAP3K1, MAP2K4, CDH1, that is the mutation that defines lobular carcinomas, RUNX1, FOXA1, and NCOR1. Whereas in basal-like cancers, we have 53 mutations, but very few additional recurrent mutations. These observations, however, confirm one important point here, that there are no highly recurrent mutations or highly recurrently mutated genes that define each one of these molecular subtypes here. For instance, PIK3CA mutated in over 40% of luminal A cancers, but it can be mutated in HER2 enriched and even basal-like. Conversely, triple-negative breast cancers are defined by 53 mutations. 90% of them harbor them. But 53 mutations can be found in the other subtypes as well. So this, at that point, let's say 10 years ago, led some to claim that these genotypic-phenotypic correlations would not actually be present in the context of breast cancer. That there's no gene that defines, or genetic alteration that defines each one of these subtypes. And that was actually something that resulted in the pursuit of completely different approaches for the genomics of breast cancer. But back in 2012, when I moved from London to New York, and with the availability of incredible archives, pathology archives, with the most fascinating rarities possible in the world at Memorial Sloan Kettering, I really wanted to understand whether there would be these genotypic-phenotypic correlations in breast cancer. And if you could explain some of these histologic special types on the basis of their genomic features. And actually, before I moved, together with Chris Lloyd and Alan Ashworth, we actually told everyone exactly what we were planning to do. We wrote a leading edge article that came out in Cell in 2011 that probably no one has ever read. But in that leading edge article, we argued that by studying these rare cancer types, we could identify several new biological features that would not be identified through these brute force approaches applied to common cancer types. And why? Because common cancer types have an incredible level of phenotypic diversity. Their genomes are unstable. They harbor many genetic alterations, and one would have to sequence hundreds, if not many thousands of these cancers to define the repertoire of somatic genetic alterations. And this is exactly what TCJ and PCOG have demonstrated. And we also argued at that time that the rare cancer types would actually provide something very different, because phenotypically they're very homogeneous. If you see one tumor of one of these rare cancer types, you see them all, they all look pretty much the same down the microscope. Their genomes are a bit of a bore. They have very few genetic alterations, but when they're present, they're usually in the form of highly recurrent genetic alterations, if not pathognomonic genetic alterations for that particular cancer type. It's therefore, by studying a small number of each one of these tumors, would be able to identify what would be their drivers. And our hypothesis was that by focusing on focused studies on each one of these types, would identify novel driver genetic alterations, novel pathognomonic mutations, novel genotypic-phenotypic correlations, and perhaps even new cancer genes. So fast forward it to 2023, we actually identified several novel pathognomonic genetic alterations. We found novel hotspot mutations in PRKD1 in polymorphous adenocarcinomas, tumors that affect the salivary glands in the breast. We found novel fusions in different types of ovarian cancer that define those histologic types. By applying this concept to a quasi-benign soft tissue lesion called granular cell tumor, we identified two novel tumor suppressors ATP6AP1 and ATP6AP2, two accessory proteins of the vacuolar ATPase, whose loss of function mutations result in accumulation of early and recycling endosomes that confer the so characteristic granular phenotype of these tumors and constitute drivers of this disease. This study was done by one of my mentees, Freja Pareja, when she was still a fellow in my lab. She's now an attending physician scientist at Memorial Sloan Kettering. And we are now exploring this concept in the context or the alterations of these genes that control the vacuolar ATPase because they control the pH of intracellular organisms. We're studying these genes now in the context of resistance to several different types of genes. And we've also identified something that we used to talk a lot about before, but we had no proof of it, that some of these phenotypes in breast cancer could be even driven by interactions between two different genetic alterations. And I think one of the most beautiful examples was done in collaboration with the group from Dana-Farber, actually, with Stuart Schmidt, and also from MGH, John Freite. We studied a tumor type of the breast that causes more controversy because of its name rather than because of its aggressive behavior. That's the tall cell carcinoma with reverse polarity. It's a vanishingly rare tumor where the cells have a reverse polarity, and rather than having the nuclei in the basal pole of the cells, they have the nuclei in the apical pole. And by sequencing these tumors and doing functional analysis, we found that they are driven by IDH2 R172 hotspot mutations or TET2 loss of function mutations, always in combination with alterations of the PI3K pathway family of genes. And by modeling this on 3D models or in 3D models, we found that that the presence of IDH2 hotspot mutations together with a PIK3CA hotspot mutation results in the acquisition of the reverse polarity that characterizes these tumors. So we have, over the last decade, we have indeed found direct evidence that studying these rare cancer types is not really a mere academic exercise. It can provide new biological insights that we could not discover just by studying common forms of cancer. But these are general examples, and I just giving you one there that was in the context of breast. So what happens in the context of special histologic types of breast cancer? And for this audience, I think it's important because I don't know how many pathology aficionados I have in the audience. So I just want to recap here what I mean by special histologic types of breast cancer. Actually, these are entities recognized by the World Health Organization as entities that are defined on the basis of their histology and seem to have different clinical features. An important point here that I need to make, and it may come as a shock to some of you here attending this talk, is that the garden variety of breast cancers, the invasive ductal carcinomas, for us pathologists, they are a diagnosis of exclusion. We call a breast cancer a ductal carcinoma when we cannot classify it into one of these 20 special histologic types. Each one of these special histologic types or rare histologic types of breast cancer account for a small minority of breast cancers, but collectively they account for almost 15% of all invasive breast cancers. They have important prognostic and clinical implications. For instance, in several studies, this comes from the Milan cohort, but from Nottingham, from Milan, from many independent groups, what we know now that even when we correct for the intrinsic subtypes of breast cancer, some forms of invasive breast cancer, special histologic types, like tubular and cribriform, they have a much better clinical outcome than the garden variety of breast cancers, ductal or lobular carcinomas. I'm not going to be talking about the clinical features of these tumors today. Rather, I'm going to be discussing with you whether they would be driven by distinct driver genetic alterations and how we contextualize this in terms of our clinical classifications now. So when I first moved then to Memorial back in 2012, the area that we really wanted to explore was the rarest of the rare. Okay, so we focused on the low-grade forms of triple-negative breast cancer. And, you know, when we think about triple-negative breast cancer, we think about aggressive tumors that are of high histologic grade, a great deal of genetic complexity. They have an aggressive behavior, but they respond to chemotherapy. These are completely different. These are tumors that are of low histologic grade. They go by rather esoteric names like secretory carcinoma, adenoid cystic carcinoma, mucoepidermoid carcinomas, and polymorphous adenocarcinomas. These tumors actually have several additional characteristics in common, that they consistently display a triple-negative phenotype. And if you profile these tumors with PAM50, they very often come back as basal-like. They have an indolent clinical behavior, and they're really vanishingly rare in the breast, but they can be diagnosed as primary tumors in the lung, and they are not uncommon in the salivary glands, and hence the name here, salivary gland-like tumors of the breast, because they're much more common in the salivary glands. We have found that these tumors have another characteristic in common. They don't harbor p53 mutations like triple-negative breast cancers, but rather each one of them, through studies from our group, from Joran Stemmen's group, and from Paul Sorensen's group at BC Cancer Agency, we found that these tumors are driven by highly recurrent, if not pathognomonic genetic alterations. Each one of them. For instance, secretory carcinomas are driven by DTV6 and TRAC3. Adenoid cystic carcinomas, as demonstrated by our group together with Britta Weigelt, again, a former mentee of mine, they're driven by the MIB and FIB fusion gene, which is a driver here, bona fide driver, present only in adenoid cystics. And if they don't have that, they have MIB-L1 rearrangements with many different partners. And if they don't have either type of fusion gene, they harbor MIB gene amplification. Mucoepidermoid carcinomas, as Fraser in my team shown, that these tumors harbor MAML2 rearrangements, the fusion genes that are again oncogenic and causative of the phenotype. In polymorphous adenocarcinomas, we've shown that these tumors, it doesn't matter if it's breast or salivary gland, they harbor the PIKG E710G hotspot mutation that's activating typical. And when they, in 72% of the cases, when they don't have it, they harbor PIKD1, 2, or 3 rearrangements. And this slide here, actually, back in the late 2010s, constituted two paradigm shifts. First, it demystified that idea that there were no genotypic-phenotypic correlations in breast cancer. Yes, there are, and you can see it here. And the second is that we have some convergent phenotypes in breast cancer. And what do I mean by that? That adenoid cystic carcinomas are a disease of MIB pathway activation, regardless of the genetic mechanism here. There are many that can result to that. Likewise, these polymorphous adenocarcinomas are a disease of activation of the PIKD family of genes. And again, regardless of the exact genomic mechanism, they are convergent phenotypes. And these observations resulted in a change in the way actually we practice the diagnostic workup of patients with triple-negative breast cancer, because the spectrum of triple-negative breast cancer has expanded massively. The majority of these tumors, okay, they are high-grade triple-negative breast, they are high-grade, most of which are actually invasive ductal carcinomas of no special type that are triple-negative. There are some special histologic types that are almost invariably triple-negative and high-grade, like medullary, metaplastic, and apocrine. And these are the ones that we think of when we talk about triple-negative in general in a study or a publication. Yeah, these are those with 53 mutations, very complex genomes, and there's even some heterogeneity within these tumors here, given that despite the fact that all of them harbor 53 mutations in over 90% of the cases, there are some that have enrichment for PIK3CA mutations or even Wnt pathway alterations. But more importantly, what studies of the studies focused on these rare low-grade variants of triple negative breast cancers have shown is that the spectrum is much wider. And we have this whole group of triple negative breast cancers that used to be diagnosed as triple negative. And they have different histology, different biology, different clinical behavior. They don't respond to chemotherapy. They behave completely differently. They don't harbor those 53 mutations. And we have shown that each one of them is driven by specific repertoire of somatic genetic alterations. And this has actually led us, pathologists, to have to histotype every single triple negative breast cancer in the clinic. And why? Because if, for instance, we have an adenoid cystic carcinoma patient with an adenoid cystic carcinoma, the therapeutic journey is going to be very different from one harboring an invasive ductal carcinoma of triple negative phenotype. In addition, if the patient has a relapse and has, for instance, secretory carcinoma, a diagnosis of secretory carcinoma now is an indication for the use of TRK inhibitors. So here we have seen how histology brought into this whole, the special types, the histology in the form of special types, has brought a completely new dimension to the way we diagnose and manage triple negative breast cancer patients. And it's not uncommon to have patients with these low-grade forms of triple negative disease discussed at our tumor boards and molecular tumor boards. But even in the context of these high-grade forms, for which we did not find the pathognomonic genetic alteration, the study of their genomics actually is helping us understand some clinical conundra that we could not understand before. So one of these rare forms of triple negative disease, the metaplastic carcinomas, okay, they are really fascinating. These are tumors constituted of a complex admixture of epithelial cells that display differentiation towards other lineages, like spindle, squamous, chondroid, osseous. These tumors are really aggressive. What we have observed by studying these tumors is that their repertoire of somatic genetic alterations is pretty similar to what we find in, let's say, unselected triple negative cancers. And you can see the results of a whole exome analysis that we did a few years back, where each column is a tumor, each row here is a gene, each square here is a mutation, coded according to the legend here. And as you can see, the most frequent mutation in these tumors is 53, and we have a few PIK3CA alterations. There's nothing that jumps here. There's no pathognomonic genetic alteration. But we found that when we compared to the garden variety, there was only an enrichment for mutations affecting the PI3K pathway genes and also the Wnt pathway genes. And when these mutations were present, by transcriptomic analysis, and here it's a big disclaimer, just by transcriptomic analysis, then we observed some activation of the PI3K pathway or the Wnt pathway based on the presence of those genetic alterations. That's okay. We've also found that there is a great deal of phenotypic, transcriptomic, and genetic heterogeneity within these metaplastic carcinomas. We found that transcriptomically, the major histologic component of these metaplastic carcinomas actually determines or is strongly associated with the transcriptomic subtypes that these tumors have. So here we microdissected the components of the tumors, profiled as RNA sequencing, or even before we did even microarrays, and observed that the classification varied according to the histologic component. For instance, the spindle component was almost invariably classified as floating low, whereas the others were an admixture of different transcriptomic subtypes. And we found as well that different components or metaplastic carcinomas with different types of differentiation harbored slightly different repertoires of somatic genetic alterations. For instance, we found that some mutations were enriched for in some of the histologic subtypes of metaplastic carcinomas than others. And we've also observed that, for instance, some third promoter mutations were inversely correlated with the presence of 53 mutations and correlated with some histologic differentiation within metaplastic carcinomas. Just to show that even within these histologic special types of breast cancer, the ones that have this phenotypic diversity, we still observe differences according to the differentiation. But going back to our original whole exome results of the metaplastic carcinomas, there was another very interesting observation that we made. When we used the whole exomes, we found that the mutational signatures, meaning the repertoire of driver and passenger alterations, when we apply non-negative matrix factorization, when classify the mutational signatures, the biological processes that appear to have shaped the genomes of these metaplastic carcinomas were two. Either they harbored an aging or clock-like signature characterized by C2T alterations, specific trinucleotide contexts, or they displayed evidence of the signature 3 or the HRD, homologous recombination DNA repair deficiency signature. 40% of the cases almost. And this was rather puzzling because work from our team and also from Paz Polak when he was working at Gary Gatz's team, we have shown that actually these HRD signatures are usually present in the presence of biallelic alterations of HR-related genes. And in the context of metaplastic carcinomas, actually, there were very few alterations that were biallelic affecting HRD repair-related genes. So we've seen a disconnect there. And the other point that actually made us stop and think whether this was actually representative of HRD is the fact that HRD cancers oftentimes respond to neoadjuvant chemotherapy, whereas, and we see this in triple-negative breast cancers as well. Up to 65% of triple-negative breast cancers respond to neoadjuvant chemotherapy. But in metaplastic carcinomas, the studies have shown that even in the combination with immunotherapy and neoadjuvant chemotherapy, responses are found between 2 to 20%. And it did not make sense that almost 40% of them were HRD, but only a small proportion of them respond to neoadjuvant chemotherapy. So what we decided to do when we did not know what to do, what did we do? We did whole genome analysis as well. So to try and understand whether this was a true HRD phenotype or not. So to do that, actually, we got 25 metaplastic carcinomas that we had sequenced before with whole exomes and did whole genomes. And we found that these cancers harbored really complex genomes. You can see here in this circles plots with a huge number of mutations and a large proportion of rearrangements, as you can see here inside the circles. These are two of the genomes of the cases. But we use this genomic information to see whether these tumors would display a complete HRD phenotype. And why? Because homologous recombination DNA repair deficiency leaves several different genomic scars that can be accurately captured by whole genome sequencing, including an increased number of deletions, small deletions flanked by sequences of microhomology, specific patterns of rearrangements, and of course, the mutational signatures 3 and 8. When you looked at only the mutational signatures, we could still see that a large proportion of the metaplastics display this HRD point mutation, the SNV signatures. Yeah. But when we integrated all of these different parameters using HRDetect, we found that it was very different. You can see here the unpublished results, but out of these cases that we sequenced, only 3 had a high HRDetect score. And then when we started digging into it, we found that these 3 cases actually had biallelic alterations affecting HR-related genes. 2 had BRCA1 alterations, and 1 had a RAD51C intragenic deletion coupled with loss of heterozygosity. So these cases displayed a complete HRD phenotype, whereas all of the others that had some degree of signature 3, that you can see here in red, yes, the red bars, they actually did not harbor the other features of HRD, and they had only a partial HRD phenotype. And we are now exploring this in greater detail. We have done some long read sequencing, and we're trying to understand what is the other type of DNA repair defect present in these cancers that leaves a signature 3 behind, but it's not true HRD. And in fact, in this cohort, the 2 patients that actually had pathological complete response were the 2 of the 3 that had a proper HRD. So we brought this story together. Another interesting observation that we made by studying these metaplastic carcinomas that I find really exciting is that when we started looking at the repertoire of somatic mutations, we found, okay, this is one example with a BRCA1 germline alteration, biallelic then, loss LOH in the tumor. And we started looking at the signature decomposition. Here we found HRD as a dominant signature, the indel signature is HRD as well. But when I started staring at this plot, I found that there were several mutations that were clustered together in close to areas with some form of rearrangement or structural rearrangement. And when we looked at the mutations, they were not the pattern that we expect in HRD. Rather, the mutations here were a mixture of C2T and C2G, so C2T and C2G, specific contexts. And they were examples of cartiges that showering of mutations close together in a region of the genome that was close to a region with a rearrangement. And we looked at the pattern of these mutations here where cartiges happened. They were all apobac. So we went back and looked at the metaplastic carcinomas, and we see the evidence of these 2 mutational processes being concurrently active in metaplastic carcinomas, which is not common in the context of the garden variety of triple-negative breast cancers. So together with Ruben Harris, we studied, who discovered the apobac enzymes, we are studying now this interaction between these 2 types of DNA repair defects or genetic instability in the context of these tumors. So another topic that I wanted to cover in this presentation is how we, by focusing on these rare cancer types, we started identifying combinations of of genetic alterations that result in a very specific phenotype in breast cancer. And I've given you one example at the beginning with the tall cell carcinoma with reverse polarity and the IDH2 R172 hotspot mutations. But the one that I'd like to talk to you now is the adenomyophthalmioma. Adenomyophthalmioma is a quasi-benign tumor of the breast. It has a very specific histology. It forms these nodules here that push the breast stroma, and they are composed of this admixture of clear myoepithelial cells and pink eosinophilic epithelial cells. These tumors have an uncertain malignant potential. They look bland down the microscope and rather cute in terms of their histology, but they can metastasize to the liver. And when you look at the liver biopsy of these tumors, it looks like the primary breast cancer, the primary breast tumor. But they can also transform to invasive breast cancer, high grade. And when they do, these tumors are invariably of triple-negative phenotype. Until we started studying these tumors, and it was Felipe Gaia and Anshi Lee in my team that we studied them, we did not know what the genetics of these tumors were. We found that unlike the different histologic special types of breast cancer, these could be ER positive or ER negative. 1/3 were ER negative, 2/3 ER positive. Regardless of ER, they look pretty much the same down the microscope. And upon sequencing these tumors, and we put a series of more than 100, finished with 40-something because we needed to have consensus for the diagnosis of those lesions. And here are the results of the analysis that actually helped us contextualize some mutations that we did not understand in the context of breast cancer before. So what you're seeing here are the results of the sequencing of ER negative or ER positive adenomyophthaliomas, where in each column you have a tumor, each row is a mutation. And if you look at here, in the ER negative, these would be triple negative lesions here. There's one gene that's missing here. That's 53, yeah? 53 is not mutated in these tumors. Rather, regardless of ER status, we found that they harbored PIK3CA mutations in a large proportion of them. In the ER positive, in the absence of PIK3CA mutations, they harbored AKT1, E17K hotspot mutations. And in the R negative, however, we had something different. We had PIK3R1 loss of function mutations. That's all great. But what we did find that was really surprising was that in the ER negative adenomyopatheliomas only, 60% of them harbor the HRAS hotspot mutations. All of them were Q61 hotspot mutations. And why am I excited with a hotspot mutation in HRAS? It's an oncogene that we've known for such a long time. It's the fact that in breast cancer, if you look at PCJ, PCOG, you don't find HRAS hotspot mutations Q61. And they are not present in the garden variety of breast cancer. So we asked the question of whether these HRAS Q61 hotspot mutations would be causative of the phenotype of adenomyopatheliomas. To do that, what we did was we used two non-malignant breast epithelial cells, MCF10A and 12A, one MCF10A with a knock-in of a PIK3CA mutation. And why did we do that? Because the HRAS mutations always co-occurred either with a PIK3CA or a PIK3R1 hotspot mutation. So we use this to study in the presence of a PIK3CA hotspot mutation. These cells are ER negative. They have the potential to buy lineage differentiation, epithelial and myopithelial cells. They lack p53 mutation. And akin to some adenomyopatheliomas that we found, and I can show you two examples here, they harbored homozygous lesions of CDKN2A to B. So they seem to be interesting models for us to test. What would be the impact of these HRAS Q61 hotspot mutations in conjunction with the PIK3CA hotspot mutation? And so upon forced expression of wild type, empty vector wild type of mutant HRAS in these cells, it did what it was supposed to do. It resulted in HRAS activation of the ERK pathway, and it's all fine. So it did what it was supposed to do. So we next asked the question of what would be the impact of forced expression of mutant HRAS in MCF10A with the PIK3CA hotspot mutation, or MCF12A cells. And upon forced expression of mutant HRAS, we saw a downregulation of icadiarin and an upregulation of pimentin. And one we could say here, oh, so this is epithelial to mesenchymal transition, but actually the cytokeratin levels were maintained across. There was no shifting in lineage here. Instead, what these cells did was to express alpha smooth muscle actin, TEMP1, and delta NP63, which actually is exactly the phenotype that we would expect in myopithelial cells. So here, what we surmise is that the forced expression of mutant HRAS in this context, and in particular in the presence of a PIK3CA hotspot mutation, result in the acquisition of at least a partial myopithelial phenotype. Upon testing this using a 3D matrigel model with these cell lines, what we saw was really exciting for me, that upon forced expression of mutant Q61R, in particular in the MCF10As with the PIK3CA hotspot mutation, what we found was that the cells started expressing myopithelial markers in the abluminal layer, like the clear cell layer in adenomyopatheliomas, and not in the epithelial cells anymore, acquiring some of the cardinal histologic features of adenomyopatheliomas. And if you look at the expression of the respective markers here, vimentin, and in this context, calponin and adenomyopatheliomas, we see exactly what we saw in the 3D model systems, which I find really exciting. And this actually has been translated into the development of immunohistochemical marker to help us with the diagnosis of adenomyophthaliomas because of the rarity of HRAS T61 hotspot mutations. We validated an antibody for the detection of T61 hotspot mutations that we use as an ancillary marker now, because it's really positive in adenomyophthaliomas for our diagnostic practice. So here I hope I have shown you an example where we identified the driver gene and how it needs to be present in the combination with another alteration, and how we translated that into an ancillary marker that we pathologists can deploy clinically. So to finish this talk, just 4 more minutes, I'd like to just to show you something that we did in the other way around. We basically, remember that I mentioned to you that one of the major problems that we have is that for the use of special histologic types in breast cancer is the fact that we cannot be really reproducible in our diagnosis. So now with the development of artificial intelligence and for the diagnosis of entities down the microscope, we decided to use this genetic information to help us develop AI algorithms for diagnosing these lesions. So, in fact, actually, despite all of our interest in these special histologic types of breast cancer, when we look at our inter-observer or category-specific agreement across pathologists, even for the histologic special types that are quite common, like lobular carcinoma, our inter-observer agreement is still modest, 78%. So we pathologists are not very good at being reproducible in terms of our diagnosis. And in fact, a study from a good friend of mine, Anva Solomon, the Formula Consortium put 35 pathologists from 9 countries and asked them to diagnose tumors from lobular to ductal, okay? And in each column here, you can see a tumor, and each row is as a pathologist, and you can see here that there are some cases that actually were consistently classified as non-lobular, other cases were consistently classified as lobular, but there were many where the diagnoses were not in agreement across pathologists. And actually, the kappa scores that are a measure of the agreement for the diagnosis of lobular carcinoma were really modest, 0.5 to 0.6, apart from the 0.8 that would be required. So we decided then to use features of lobular carcinoma for us to develop a solution for the diagnosis of lobular carcinoma. Lobular carcinomas are the most frequent histologic special type of breast cancer. They are characterized by this this discohesive monomorphic cells that invade the breast stroma as single cells or single cell files. They're quite monotonic in the way they look. And many investigators and confirmed in our large-scale study from Memorial Sloan Kettering is that greater than 70% of these tumors harbor biallelic inactivation of CDH1, the gene that encodes for ecadherin. There's in the context of breast cancer, biallelic alterations of ecadherin, CDH1, are pathognomonic for lobular carcinomas, but they're present in 70 to 80% of the cases. Ecadherin, the gene encoded by CDH1, is an addition molecule that mediates homophilic and homotypic additions. So for 2 epithelial cells to stay together, they require normal levels of ecadherin, and it is the very loss of ecadherin that causes the so characteristic discohesiveness of lobular carcinomas, as demonstrated by Jos Jonker's group and many others. And this can be caused by biallelic mutations, homozygous deletions, or promoter methylation. So then what we did was, we looked at all of our MSK series, and we found that CDH1 mutations, biallelic mutations, are present in less than 0.3% of other forms of breast cancer. So we reasoned, perhaps we can use the biallelic information for us to train an artificial intelligence algorithm applied to histologic images to diagnose lobular carcinomas. So at Memorial Sloan Kettering, we routinely digitize every single whole slide image. So we basically then, together, Freja Pareja in our team, basically put together a large series of breast cancers where we had MSK impact data, our multi-gene panel, where we could define whether there were biallelic mutations of picadirine or not. We had the whole slide images. Then we trained an artificial intelligence algorithm to detect CDH1 biallelic mutations or not. And we were hoping, that was our hypothesis, that it would be diagnostic of lobular carcinoma. In no point during the training here, we used the information lobular carcinoma. So the training was done in a very simple way. We split the slides into tiles. Then we extracted the embeddings in the vectors using ResNet. And then we used a multiple instance learning system that we developed in partnership with Paige to diagnose invasive or pre-invasive breast cancer. And then we used a very simple forward aggregator just to say, is there a CDH1 biallelic alteration or not? And we did this in a 3-2-1 training, tuning, and testing. And then we applied this to 2 additional data sets. And let me show you the data sets. We had the MSK development data set, then the MSK validation data set, and then we use the TCGA data set for final validation. And to cut a long story short, what we found is that this algorithm could identify indeed CDH1 biallelic alterations with an area under the curve of 0.96. And actually, here are the results there under the curve with a pretty decent precision recall, but there were some false positive cases, meaning cases that were wild type by sequencing that were not biallelic. They were not wild type, they are not biallelic, but were classified by the AI as lobular. So we went to these cases, and what we found is that they did not have biallelic, okay, but they actually instead had gene promoter methylation, or they had intragenic deletions that were not originally detected in impact, or they had novel fusion genes that were loss of function fusion genes affecting CDH1. So the algorithm actually brought to our attention additional mechanisms of CDH1 inactivation that we actually had missed. When we applied this algorithm then to diagnose lobular carcinomas, this is in the validation cohort, we had an area under the curve of 0.98. And this is far better than 78% agreement that we get by pathologists. And we had some cases that were misclassified by the algorithm, and these were not typical lobular carcinomas. They were actually cases that actually were either solid lobular carcinomas or mixed tumors where there were lobular areas and non-lobular areas. So you could even differentiate these cases, but did not classify them as lobular carcinomas. But this is such an improvement against what we do in our diagnostic pathology practice. And we went there from genetics to a histologic diagnosis without using any annotation from pathology. And this is another validation data set, just showing that, again, the area under the curve is way above the 0.9 mark. So in conclusion, I hope I have demonstrated to you in this talk that by studying the genomics of these rare cancer types, so we could identify novel genotypic-phenotypic correlations, novel driver genes, then we could actually refine the way we classify and manage invasive breast cancers. And I think a message for the whole group here is that just because something is rare, it does not mean that it's uninteresting. It doesn't matter if the editors of the journal say, oh, you're just studying a rarity, it's not of interest to us. Actually, studying these rarities can be incredibly informative, and they may constitute the exceptions that explain many rules that we cannot understand by just studying the common cancer types. With that, I'd like to thank you for your attention. I have many people in our team to thank, and it was just a great pleasure to be here with you today. Thank you.
Thank you very much, Doctor. That was really interesting. And we are open to all your questions. Please type your questions in the Q&A section. And actually, we have our first question from Ashish, and he wants to know, since you didn't use any supervised not labeling annotation, what was the training technique to do this?
It's a great question. So what we did was, first, we had 40,000 breast cancers, completely unrelated, where we did multiple instance learning analysis only with labels. Normal, pre-invasive, and invasive breast cancer. We did this using multiple instance learning with a CNN and an RNN, as described by Thomas Fuchs in his Campanella et al. Nature Medicine paper in 2019. So we use that to do the embeddings and vectors, and then we use these embeddings and vectors and put through a forward aggregator neural network just with the labels, CDH1 biallelic or non-biallelic. The trick was the first step, the middle one. Without that part of the classifier, this would not have been possible.
And I was wondering, for these genotypic phenotypic correlations, did you find some correlation between the genotypic and the immune system, and how do they correlate it?
Yes, I did not have time to go into the details, but Nadim Riaz, in our in a study that he was the PI, we have a Star Cancer Consortium brain together to further exploit this. But we found, for instance, even in the context of breast cancer, BRCA2 and BRCA1 have fundamentally different impacts on the amount of new antigens and the immune responses. And we could map back to the increase in these deletions flanked by microhomology, particularly in the BRCA2s that were orders of magnitude higher, and more new antigens in the BRCA2s compared to BRCA1s. And then he did models, and again, only models, syngeneic models, where he inactivates BRCA1 or BRCA2, and he saw differences in response to immune checkpoint blockade using the 41 model and another model. Now, we have some retrospective data from patients treated with immunotherapy at Memorial that suggests also an interaction there between, let's say, different genotypes or different types of DNA repair defects caused by biallelic inactivation and different immunotherapy responses. But again, it's only retrospective data.
Thank you very much.
Samstein et al., Nature Cancer 2021.
Thank you again. And well, I think we don't have more questions, but thank you for your talk. I really enjoy it. I really enjoyed to get to know you and get to hear your talk.
Thank you. It's a great pleasure.
I think that will be all.
Thank you.
Good. Thank you. Have a good day.
Bye-bye. Bye-bye.